Thanks, Tiffany. Thanks, everyone. So what I want to talk to you today about is a project that we've been doing for a couple of years now, looking at thermal tolerance and mechanisms of thermal tolerance in corals on the GBR. Okay, so I know for this audience, going through the details of global warming is a bit of a given. Temperature has increased worldwide. It's projected to increase more. Sea surface temperatures on the GBR are estimated to increase 0.1 degree per decade. 50% of the projected warming and the warming that's going to happen in the coming decades is now considered unavoidable. So what that really means is for corals on the GBR today and those reefs living today, there's no escaping temperature change. For those corals, they will have to increase thermal tolerance or the tolerance to temperature change to not just to survive these changing thermal conditions, but to be able to reproduce under changing temperature conditions. And this is going to be vital for future intergenerational change and adaptation to deal with further climate change. So really what that tells us is the capacity of corals that are living on reefs today to change their physiological thermal tolerance is crucial to future survival and stability of the reef. So the project that we've been running over the last couple of years is to look at the mechanisms that will determine if those corals of today can win or lose under climate change. And so I want to start out by saying that this is very much a cross-discipline study, and it has relied on the expertise of a lot of different people, all the way from looking at SST profiles historically on the GBR through to the coral stress and algal stress responses, and also working with Pete's group to look at the implications for this research to corals on the GBR. So the first question we've really asked is, what are the short-term temperature patterns of past thermal stress events on the Great Barrier Reef? I particularly mention short-term temperature patterns because we know it's not just the minimum and maximum temperatures that organisms are exposed to that are important in determining their tolerance to temperature and temperature change, but it's the variability and those short-term variations in temperature which really influence how, whether an organism can survive. So if we look at how bleaching is discussed in the literature and how it has been discussed, the sentence on the far side, variations of this can be found in pretty much any paper on coral bleaching, and that is that coral bleaching is caused by a sustained SST or sea surface temperature increase of just 1 to 2 degrees above long-term summertime averages. And I think if I asked most of you if this graph is a fair representation of the thermal environment that corals are exposed to coming up to bleaching, we generally say yes, that is what corals experience coming up to bleaching, which is an increase in temperature up to a bleaching threshold, and then bleaching occurs. But what's interesting is when we've looked back at temperature patterns over the past 30 years on the Great Barrier Reef, this really isn't a fair description of the thermal environment that corals are mainly living in coming up to bleaching events. In fact, when we've categorised the stress events across the reef, so you recognise the Queensland coast here, these pixels are 50 square kilometre grids of the satellite data. So 74% of the past thermal events that corals have been exposed to on the GBR are characterised by a sub-bleaching temperature pulse that the corals are exposed to before bleaching conditions. So you see we've got 3 temperature profiles here, coded blue, green and red, and this reflects the colour coding of the pixels on the side. So in the top you'll see a trace of temperature coming up, exceeding the maximum monthly mean, and a period of a low temperature stress, followed by a relaxation period in which the temperature goes back below the MMM before corals are exposed to the local bleaching threshold. And that's what we've seen predominantly in the SST profiles of the past 30 years. Far less frequently, we see temperature come up, exceed the local bleaching threshold, followed by a relaxation period where the temperature cools off again, and then exposure to a second bleaching pulse. And then finally, less than 20% of thermal events, we see that more kind of classic description of SST going up and breaching the local bleaching threshold. So here's just a decision tree just to help understand how we've characterised these SST patterns. So basically, does the temperature exceed the local bleaching threshold? Yes or no? Yes, then there's a stress event. Does a pulse occur? Yes or no? If no, that means that the temperature has exceeded the MMM and it's not followed by any relaxation period before exposure to the bleaching event. If yes, does that pulse exceed the MMM? So is there two bleaching temperature level pulses? If yes, we characterise that as a repetitive bleaching trajectory, and that's how I'll refer to this temperature trace through the rest of the talk. If no, so there's an exposure above the MMM, a relaxation period, and then bleaching exposure, we refer to this as a sub-bleaching trajectory. So we've categorised these three temperature traces based on exposure to bleaching level temperatures. And we can break this down across the GBR. So again, you've got the Queensland coast, and we're looking at the frequency of these temperature trajectories in the historic or past 30-year satellite data. And what you can see is across the GBR, the predominant exposure of corals is to this sub-bleaching trajectory in blue, whereas if we compare it to the direct bleaching trajectory, this is very rare on reefs and occurs very rarely. What are the drivers of this kind of short-term temperature pulsing? Well, we know wind and current both cause a mixing of ocean waters, and this is where lower, cooler temperature water is brought up to the surface and mixed with warmer surface water. And lunar tidal cycles are linked to this kind of SST pulsing across the continental shelf and in lots of different environments. And we also know neap tide conditions, so the slack in the tide is associated with bleaching conditions due to that reduced mixing. And so with a tidal range on the GBR of around 3 metres, it's easy to see what's driving this kind of regular temperature pulsing. Tide is not considered to change under future climate change. And so given that, we've wanted to look at what's the potential SST profiles that corals may be exposed to in the future as temperature goes up. And how we've done this is we've applied temperature offsets to the historic SST profiles, and we've done this all the way along the grid of the GBR with temperature offsets from 0.1 degrees Celsius up to 2 degrees Celsius. And we've just got them grouped here with the plus 0.5, plus 1, plus 2 degrees Celsius. And what you can see is as temperature goes up, the frequency of the sub-bleaching trajectory begins to come down, and the frequency of a direct bleaching trajectory goes up. And this is due to the loss of that relaxation period below the MMM as temperature starts to come up. So that pre-exposure starts to disappear, and we start to see a continual build-up of temperature. And if we break this down across the Queensland coast, here you've got, again, current plus 5, plus 1, plus 2, just to show you that there, what we would see is a potential GBR-wide shift away from temperature profiles that expose corals to these sub-bleaching pulses, and more towards the direct and the repetitive bleaching trajectories, which become more frequent. Why does this matter? What would this mean to corals? So we know, as I said before, temperature exposure is really important in determining an organism's thermal tolerance. And when we talk about thermal tolerance, what we mean is it's the temperature range at which the organism's cells can minimize or prevent irreparable cellular damage. So it's what determines the temperature range in which an organism can survive. And this obviously then influences the organism's ecological distribution. So obviously the next question has been, well, what does this really mean to corals that are living in these, living and experiencing these temperature profiles? So we've looked at the coral and algal stress response under these different temperature profiles and looked at characteristic markers of heat shock, cellular stress, antioxidant production, cell death and cell survival in response to these temperature conditions. And this is just to show you, we've replicated these temperature trajectories with corals around Heron Island, exposing them to repetitive bleaching trajectory, direct bleaching trajectory, and the sub-bleaching trajectory. We've also included a whole range of different controls, which I'm not going to go into today, but if anyone's interested, I can show you later. So what I want to show you here is, first of all, one of the classic measures of bleaching is to look at the algal photosynthetic yield. Now this is a measure of bleaching. It shows us the breakdown of the coral-algal symbiosis. So if we follow the yield through the exposure to these temperatures, we monitored all the way from the pre-stress period, recovery, and throughout the second stress. What we can show is that regardless of pre-stress, corals undergo bleaching. So under all those three trajectories, corals will undergo a bleaching event when exposed to those bleaching thresholds. But what's really interesting is when we look at the animal host response under these different temperature trajectories. And what we find, if we look at, for example, the host heat shock protein expression, this is a classic marker of heat shock. We find that corals that are exposed to this sub-bleaching trajectory have significantly lower HSP expression during coral bleaching. Now, we didn't find this was the case for antioxidant production, but what was very interesting is that we found significantly lower expression of the genes that regulate cell death in corals that have been exposed to the sub-bleaching temperature trajectory. So you'll see here, which is coming up as orange and green, the corals that are exposed to the direct and repetitive bleaching trajectories have similar levels of expression of genes that are involved in both the inhibition of cellular death and the promotion of cell death, but those that have been exposed to that sub-bleaching trajectory have significantly lower levels of expression of both inhibitors and promoters of cell death. So what does that tell us? So sub-bleaching exposure doesn't prevent the breakdown of the symbiosis or coral bleaching, but it does minimize HSP expression and cell death regulation during bleaching. What does this mean for coral survival of bleaching? So here what you're seeing are tissue sections of corals that have been exposed to these different temperature trajectories. In the top blue, the sub-bleaching trajectory, the middle green, the repetitive bleaching trajectory, and down the bottom, the direct bleaching trajectory. And hopefully what you can see is in the top, there's much less red stain. The red stain stains dead cells, blue is cells that were live. And so what you should be seeing here is in the green and the red, we're seeing a lot more red staining of the cells. So if we look at the proportion of cells that are live and dead that have been exposed to these different temperature trajectories, it's quite interesting. So under the sub-bleaching trajectory, even though bleaching has occurred, only 30% of the coral host cells are dead in the bleached tissue. However, corals that have been exposed to a repetitive bleaching trajectory or a direct bleaching trajectory have far more cell death. So under the repetitive bleaching trajectory, around about 70% of the coral host cells are dead during the bleaching event, and we see a lot of tissue breakdown. Corals that have been exposed to the direct bleaching trajectory, around 55% of the coral host cells are dead associated with that bleaching event, and we can also see evidence for tissue breakdown. So what does this tell us? Even though a sub-bleaching exposure doesn't prevent coral bleaching, it does minimise the impact of the bleaching event to the coral host. So that tells us is that that type of temperature trajectory where corals have a pre-stress, a relaxation period before bleaching, the coral host is able to minimise or prevent irreparable cellular damage in response to temperatures that cause coral bleaching. But unfortunately, that's also shown us that under direct and repetitive bleaching trajectories, the bleaching event is far more physiologically damaging to the coral host. This is a bit of a concern given as we look at potential for SST increases across the GBR, we see that this sub-bleaching trajectory starts to decrease and that we're looking at a greater potential for a greater frequency of more physiologically damaging SST profiles across the GBR than has been seen so far. And this has some real potential for biological and ecological implications. So if we think about for example, just comparing the repetitive bleaching trajectory and the sub-bleaching trajectory, what does that 70% of cells versus 30% of cells dead mean to the organism? If we think first about capacity to maintain heterotrophic feeding under those bleaching conditions, corals that have been exposed to the repetitive bleaching trajectory, the polyp structure is damaged and the tissues are broken down, so they're far less likely to be able to maintain heterotrophic feeding compared to, for example, the sub-bleaching trajectory where the polyp remains intact. Colony recovery from the bleaching event is also probably likely to be slower. If you think about 70% of the organism being dead, recovery would be reliant on things like regeneration, proliferation of cells from the remaining cells that have survived, remodelling, regrowth, and symbiont uptake, whereas where the tissues remained intact, recovery would be really driven just by symbiont uptake. There's also potential for disease susceptibility to be far higher in corals that have had a greater degree of death in the colony. There's less tissue to prevent microbial colonisation and to elicit immune response. And damaged tissues mean more sites for algal growth and opportunistic pathogen invasion. And this would have potential impacts on colony mortality, with greater colony mortality under a repetitive bleaching trajectory. And the flow on from that to reef recovery means that under a repetitive bleaching trajectory, we're likely to see recovery become more reliant on recruitment and repopulation rather than predominantly colony recovery from the bleaching event. So to look at this in more detail, you're probably all aware of a paper published in 2014 by Pete Mumby's group and with Juan Ortiz as the lead author, and they've been doing some modelling of coral cover across the GBR under climate change. And so working with them, we've used a percentage of dead cells under these different trajectories as a proxy for colony mortality associated with bleaching. And they've incorporated this into their models of coral cover into the future on the GBR to see what kind of implications this may have. And so what we're seeing is that under a sub-bleaching trajectory, coral cover losses are potentially slower with a greater capacity for recovery. However, under the more physiologically damaging temperature trajectories, there's the potential for coral cover losses to be much faster and recovery of those reefs to be much slower. So in conclusion, what did we find in this study? So firstly, that the thermal stress events over the past 30 years on the GBR have really been dominated by sea surface temperature conditions that expose corals to a pre-stress or a temperature exercise before the bleaching event. And that this temperature exercise, this sub-bleaching exposure, reduces the impact of the bleaching event to the coral animal. And that there's the potential that future increases on the GBR, future temperature increases on the GBR, could alter these temperature profiles away from ones that confer thermal tolerance to corals towards profiles that are far more physiologically damaging. And that this could have large implications for coral survival and coral cover on the GBR. And I just want to thank everyone who has worked, put a lot of work into this project over the last few years. It's definitely been a large collaboration and done really well.